Let me go ahead and press the record button, so I'll have an audio recording of this class. I don't think you'll get as much visually. You'll be able to follow along with the slides. But let's go ahead and get started. We're behind schedule, so we're going to get into some basic neurology this week, talking on a very basic level on how people and humans function. All right. So a lot of this PowerPoint I borrowed from Dr. Davis, but took a lot of it and made it my own. I want to follow up from last week. We'll get started with a video. This will basically say what I said in 3 hours in about 5 minutes. So hopefully it'll give you kind of a nutshell of what we talked about.
Underneath the newfangled cortex, the brainstem, the limbic system, and the basal ganglia date back to the mastodons, the dinosaurs, and the first amphibians. In humans, the old parts of the brain oversee emotion and help build memories. They control heart rate and breathing. They also form intimate connections with the new.
Who did I lose? I lost somebody and I lost the audio. I guess Renee didn't show. Jennifer's still there. I see Jennifer. All right. Now my wow, that completely messed this up. Hold on, let me stop my content and start it over again. Was the audio coming through on different locations?
Yeah.
Yes.
Yeah.
Okay. Okay. Good deal.
These checks. It filters and orders the outside world for us and allows us connections with the new brain, the cortex. Though less than a quarter-inch thick, the cortex is the brain's crowning glory. Among its roles, the cortex is our reality check. It filters and orders the outside world for us and allows us to see, touch, hear, and speak. The cortex is also the human thinking cap. All our plans, thoughts, and ideas originate in this layer. The cortex is packed with nerve cells. About 2/3 of all our neurons operate here. A piece of cortex tissue no larger than a pinhead can house 30,000 of these cells. Each neuron has a job to communicate with other neurons. The brain works by forming networks among these cells. The long, spiny branches of the networks create a neural forest of astounding intricacy. Neurons use these communication lines to talk to each other with electric and chemical signals. Here under microscope are two neurons linking up. Though it may look like they fuse together, neurons don't actually touch each other. A closer look reveals that a tiny gap called the synapse separates their branches. This is where a message passes from one neuron to the next. The message comes from here, small sacs that store chemical molecules. When stimulated, these sacs release their molecules which cross the cell membrane into the synaptic gap. An electric zap allows this to happen. Meantime, the receiving neuron has special welcome sites for the incoming molecules. These receptor sites bind with the molecules. When they do, special gates open up. The gates let in a flood of charged particles, sodium and potassium ions, which start up a new electrical signal in the receiving neuron. This simple chain of events, an electrical zap, followed by chemical changes, followed by another electrical zap, is the basis of all brain activity. It's how neurons speak to each other. Here, then, is the key to the brain's complexity. There are 100 billion neurons in the brain. Each neuron, like a bit player in a grand production, processes its information, then hooks up with as many as 50,000 other neurons to send and receive messages. 100 billion neurons times 50,000 connections. It's this complexity that allows us to think imaginatively. On their own, neurons aren't very bright. But put 100 billion of them together in a small space and let them all start talking to each other, and you start to get brainstorms. The trillions of neural networks, like an improvisational orchestra, create new ideas and connect different thoughts in a whimsical and sometimes inspired fashion. It's this impromptu ability to produce new things in our brains that allows us to progress, that quite simply takes us from here to here.
Okay, so thoughts and observations on our little clip that we watched. Anybody struck by anything? Anything different, new?
I like the way it showed the information, the zaps going from one to the other.
Yeah, I didn't quite get there. I didn't have enough illustrative talent to get there. So yeah, I did enjoy. I wished I'd had that last week at the end of class to kind of sum up everything. It didn't get into the addiction part, but it definitely got into neurotransmission, which is what we're going to talk about tonight. Did anybody catch it got cut off because we lost OPSU? Did anybody catch the thickness of the cerebral cortex?
Less than a quarter inch.
A quarter inch. So, you think about the pictures kind of betray, looks like those are really thick tubes that are up there, it's actually very narrow band, a quarter inch thick. That doesn't separate us that much from the other animals on this planet. If you think about it, you took the last scene there where it showed the cave and then shows these great skyscrapers. Evolutionarily speaking, we're not that far removed with that quarter inch of cortex that's up there. So that's something to think about. One thing, if folks have their textbooks, I will reference our text a few times tonight. If you're into reading the text, which I wasn't so much as an undergrad, I like to hear what the professor had to say. If you look on page 70, and under I'll give everybody a chance to. I'll turn the sound down a little bit too. Somebody's back. Something happened. I didn't do it. Ponca City's there. Oh, it's OPSU again. Renee, are you there? Nope. Alva, are you still with us?
Yes.
Okay, thank goodness. I didn't see. I was like, okay, they got lost in cyberspace again. Okay.
Renee isn't coming tonight.
Okay, thank you.
So that's why I disconnected from it.
Okay, no worries. We're good. We don't. If she's not coming, we're good. Thank you.
Okay. Yep.
Brain. It's under brain on page 70 at the bottom. If I could have somebody to read that for me, please. How about Rhonda? You're kind of up front there, and Alva. Do you mind reading that?
No, it's fine. It has been estimated that the human brain contains 10 to the 11th power 100 billion neurons. On average, each neuron has synapses on 1,000 other neurons and receives an average of 10,000 synapses. For many years, it was. Do you want me to keep going?
No, that's good. That's good. So
Okay.
is there anything different about those numbers than what we just heard in our video?
The numbers are
Brooke, what were you going to say?
quite a bit different.
There's quite a bit of difference there. Each neuron has synapses on 1,000 other neurons and receives an average of 10,000. So what do we say? The video that we just watched said 50,000. Notice the date. I want you all to become aware of how we cite studies. What date is that study that they referenced in your textbook?
1985.
1985. So pay attention to the 50,000. You know, I said 5,000 last week. That was research from 10 years ago. So again, we get more and more advanced as we get more and more technology. So you won't be tested on that number because that number is in flux. Apparently, we don't quite know. Anyway, so
5 years ago it was 5,000.
10. Now I'd say more like 10 years, probably roughly 15 years ago. The number was at estimated to be 5,000 per cell. And you just heard this video that we watched, you know, from a couple of years ago, estimate 50,000 per cell. So there's a lot of things. Regardless, there's a lot of things happening on top of your heads as we go through this. So anyway, that's a reference for you. Let's get into neural anatomy a little bit and make sure that Enid can see what I'm doing on the computer here as well. I do like to move around some. I'm going to move this back a little bit. Okay, so oh, stop. So we're going to go over some basic geography of the brain, topography, try to understand what structures are in place, and then we're going to kind of put my cerebellum is not helping me right now. Actually, I feel pretty coordinated, but I don't look coordinated on running this thing. So what we're going to do is we're going to help put names to the different areas of the brain. This is going to be very important to you because at the end of the day, like I said last week, I want you to begin to think about, you know, when you see behaviors with people, okay, what area of the brain is causing that behavior? What area of the brain, if I'm treating them, if I'm doing a medical procedure with them, if I'm doing counseling with them, what area of the brain is misfunctioning or is dysfunctional in this child or this adult? You know, does it have to do with their amygdala? Does it have to do with their hippocampus, their memory? Does it have to do with their hypothalamus? Are they dysregulated? And so I want us to start, does it have to do with their corpus callosum, which integrates the two halves of our brain together? I want us to start thinking about that because that's where we're at in our science. That's if you go on and get a doctorate, that's where you'll be going, is trying to understand. And we're so advanced in our imagery, our ability to map the brain and understand what's happening. We're way far, just 20 years ago, we're way past that. So we're going to get into a little of the technologies that help us understand what's going on, but we need to be able to have some common language to know what's going on too. So anyway, so what we've got here, three basic parts, and you saw these last week in my illustration. A brain stem, back, and won't go into that too much, the lower part of your brain. Cerebellum, which literally translated from Latin is little brain. And cerebrum, which is your cerebral cortex, meaning brain's bark. You know, what's the most important part of a tree to get its nutrients to the tree? The brain's bark, yeah, or the bark of the tree. So, you know, that's, you know, if you're talking about your cerebral cortex, for humans, that's a very important part of our brain to make us uniquely human. And there I've got them labeled for you again. So you should be able to recognize those on any exam. All right, so stepping into a little bit greater detail now. And this is on page 71 for those of you that want to follow along in your book. There's an illustration down there at the bottom figure 410. I really like to put pictures with words, but this kind of gives you the name of the part of the brain and also what it does. So we mentioned medulla oblongata briefly last week, but you have breathing, sneezing, coughing happening, way down here. Very basic, if you don't breathe, you don't live kind of thing. And think about a little baby. Even a little baby that's first born can cough and sneeze. That's online. That's wired up. So that's really basic stuff. Pons is a bridge. And so you see this kind of, looks like there's tendons and it looks like ligaments coming across here, striations. That actually wraps around your brainstem and connects with your the cerebellum, which is back here, which controls movement. So pons is a bridge, literally mean bridge, connects spinal cord and the cerebellum, helps you move. Thalamus. Here's our thalamus. That's that big red part that if you go from last week, it's the upper part of this brainstem. It's basically your sensory switchboard. It's helping your senses get routed to the proper parts of the brain, like vision here, your occipital lobe, your senses of touch up here, parietal lobe. Hypothalamus. I put in the 4 F's. The hypothalamus is underneath, and you can kind of see it. It looks smaller than the thalamus in the bottom of your text, page 71. It's not in color, but that's controlling the 4 F's: feeding, fleeing, fighting, and sex. So anyway, that's kind of your drives, your basic drives, basic functions there. Your pituitary gland. I expected more laughter on that when I put that together. People are asleep, so that comes from my undergraduate professor too. I didn't make that one up. I have to give Dr. McDonald some credit there. Pituitary gland is your master gland. We're not going to cover that too much. Right, kind of in the center of your brain. Reticular formation, which isn't covered here, but it's deep inside. It's inside here, and it has to do with arousal and sleep. Raphe nuclei below the reticular formation. It's involved with sleep as well. Your colliculi, which do a lot with vision. Here you can see those here, right below the pineal gland, the superior colliculus and the inferior colliculus, and those have to do with vision and auditory processing. I put synesthesia up here when we talk about LSD and some of the hallucinogens. Some of those transmitters, some of those connections are very close and spill over, and so you have that combining of those sensory perceptions. Okay, more on the brain, and this is just kind of some general information. You have your reticular activating system. So you'll notice some systems. When I say system, it's more of a combination of structures in the brain. It's not necessarily isolated to just one, like hypothalamus, thalamus. Reticular activating system has to do with wakefulness and Raphe system sleep. Helps balance emotions and reasoning parts of the brain. GABA inhibits this portion of the brain and is what barbiturates induce, a GABA release. So you think about GABA also being involved in like Ambien, Ambien for sleep. So that's acting a lot on the reticular activating system, helping people doze off. The locus coeruleus has a lot to do with mood, fear, panic, anger, depression. There's more GABA release there. Generally, GABA is an inhibitory neurotransmitter. We're going to talk about that later tonight. Basal ganglia, voluntary movement. There's actually dopamine involved in the basal ganglia. It's not part of the mesolimbic dopaminergic system, but we are looking at another role of dopamine in voluntary movement. And so, if anybody's seen, there's a movie with Robin Williams. Oh gosh, was it Awakenings? Yes. Who said that? $64,000. Is that Shayna? Sarah? Sarah, is that you?
Yes.
I want to make sure I give the money to the proper source here. So, okay, yeah. What was going on in that movie, Sarah?
Robin Williams played a doctor, and he was just seeing patients for like schizophrenic conditions, and he was experimenting with L-Dopa.
Mm-hmm. That's correct. And so he found that by administering L-Dopa, he was able to improve these Parkinsonian conditions. Now, what went bad is eventually, it was Robert De Niro who did a great job playing the patient in that role. But eventually, the brain got used to L-Dopa and the Parkinsonian symptoms returned. So anyway, dopamine does more than one thing too. I made it sound last week like it is one thing with addiction. It has several roles, like, because there's only 50 chemicals in the brain. Oh, okay, sorry. Limbic system, fight or flees, rewards, reinforcement center, mesolimbic dopamine system, hippocampus. So again, when we're talking about addiction, we're going to get into the limbic system quite a bit. Hippocampus has to do with learning and memory. Amygdala has a lot to do with anger. If you look at the cortex, again, that's the bark, that's the higher brain functionings. Glutamate and GABA are primary neurotransmitters for here. And we talked about teratogens, again, being substances that are very harmful to fetus. Okay, moving along to page 72. Here's your cerebellum. It's right back here at the base of your brain. The cerebellum helps provide smooth, coordinated movement. So, if you're putting on your high heels, or that's what's helping you. That's what's helping you move around. It's what's helping you take notes right now and write down and coordinate your handwriting. Voluntary actions are planned and initiated by parts of the cortex and basal ganglia. Remember, we just said that was involved in Parkinson's, and sent to the cerebellum, where they are integrated, coordinated, and refined into fluid movements. So this then, coordinates with the pons and goes down to your spinal cord and coordinates your muscles.
So what does that say about those of us who are clumsy and walk into walls?
It's interesting. They've done studies, a lot of studies on the influence of cortisol and brain development, and cortisol actually impacts. There's a couple of other structures that are really important in coordinating movement, and cortisol can really affect those. And we're going to get that to that point in the semester. So, thank you. You foreshadowed a.
I was just curious because there's some people that are just so ungraceful in your life.
No, there's probably a biological reason. We're learning more and more. There's a structural reason a lot of times for that. Oh, I had a nice little graphic that I had integrated there, but so that's the cerebellum. Okay, now let's talk about brainstem, cerebellum. Now let's go to the third part, cerebral cortex. This is really where we come up with our complex cognitions, just our ability to analyze, our ability to be human. Frontal lobe, we're going to talk about our prefrontal cortex, which is the best of our best. The frontal lobe is our best. It has a lot to do with our reasoning, our thinking ability, just very quickly. Occipital lobe, generally speaking, controls your vision. So, you take vision, information comes in from each eye, it crosses over, actually, and so information from your right eye is actually being processed in your left hemisphere. Information from your left eye is actually being processed back here in your right hemisphere. It's why if you have somebody that's been through a stroke, you'll often see impaired movement or impaired abilities on the opposite side of the brain where the stroke was, or on the opposite side of the body from where the stroke was in the brain. Parietal lobe in the very top, it has to do with association. And we'll talk about that in just a minute. Temporal lobe bottom has to do with hearing, speech, and vision. Wernicke's area and Broca's area, specific to speech and hearing. A little bit of vision in there too. You'll notice here I have a primary motor cortex and a somatosensory cortex and a central sulcus. You know, there's one dividing line here in between those. That's an important kind of part, because when you're looking at those two areas of the cortex, it's very interesting, because one has to do with your sense of touch, with the information that your body's so if you, like, everybody take your right finger and run it along your left arm like that. And so what's happening, just like that vision, it's getting processed up here going through, and it's being processed on this side of your brain. If it's on the left side, it's on the right side of your brain. But there's matching places, there's matching areas in the cortex that match where that is on your body. And so what they've been able to do is they've actually been able to, and I've seen the illustrations of this. They've actually been able to, because you know your brain has no sense of touch. Your brain has no sensory neurons. It is your processing. So if somebody had your skull, you know, removed, part of your, we're not talking about Hannibal Lecter or anything like here. If somebody had part of your skull removed and somebody was poking on your brain, you could not feel that. There are no sensory receptors to your brain. It is the sensory receptor. So anyway, so let's say somebody is stimulating, somebody has an electrode and finds a group of neurons and stimulates those. You would literally feel that, and if they did the right ones, they stimulated the right neurons, you would feel that same sensation going down your arm, even though it wasn't really happening. So you can, you know, falsely stimulate that. So on this slide here on letter A, it says somatosensory cortex. You can see that there's, you know, there's like the mouth, the face. There's a big section for your face. Your face has a lot of sensory, you know, there's a lot of sensory receptors. You know, this is a very sensitive area of your body, just kind of like your fingertips too. So you see a large section of the body, of the brain devoted to sensing sensations from that area of the body. You see not so much on the back. Sometimes it's hard, you know, people will like trace like numbers and letters on your back, and I can't really recognize that because I don't know what that is, because you don't have as many sensory receptors back there. Notice there's a pretty big area for the genitals. And you notice those are real close to the feet there too, which is very unique. So you have the somatosensory cortex, and if you move over here, you have on the other side of the central sulcus, you have the motor cortex. And so this is where if you're initiating movement, now if you think about that right hand that came over there and drew on your left forearm, that right hand is being controlled by the motor cortex. Same thing would go true if you hooked up an electrode, somebody stimulated that area, or now be on this side, somebody stimulated that area of the brain, you would get involuntary movements, even though you see what I'm saying? Now, they've actually done experiments like this where they've taken people who were awake and stimulated their brains while they were in a wakeful state, and then they would take notes of what they were experiencing. So very interesting, very interesting kind of things to think about there. Does that make sense? Okay, thank you. All right, so let's move on. This is on page 74 and 75 in your text, and there's a good illustration on 75. It's not color, but it does its job. The limbic system is a group of structures that surround the brainstem and form a border between the stem and the cortex. And these are some really important when we're talking about controlling human behaviors. We're talking about impulse control in kids, talking about people becoming aggressive. These are things that we deal with a lot in psychology. If you're in practice, people generally don't get referred to you because they're functioning fine. They get referred to you because they're having some dysfunction. And a lot of times it has has to do sometimes with violent behavior. Sometimes it has to do with impulse control, some of these things. So these are involved in memory formation and emotions. If you look in your brain, or on 75, look at the picture of the brain there. You'll notice, if you'll find the hippocampus. Everybody find that in the figure 411 on page 75. If you find the hippocampus, looks like it's just above the cerebellum there, and right next to the amygdala. The hippocampus, it literally means seahorse, is very, very influential in memory formation. People that have suffered damage to this have a hard time making new memories. If you look at children who have been exposed to a lot of traumatic experiences growing up and have had a lot of cortisol, cortisol disrupts this area of the brain, the development of this area of the brain. The cingulate gyrus is part of the limbic system, and the amygdala as well. The amygdala, literally meaning almond, has a lot to do with emotions, particularly very stressful and negative emotions, such as aggression, fear, and rage. So if you're thinking fight versus flight, in this limbic system, you know, it's kind of activating us. It can activate us. More structures. Let's keep going. The thalamus. Again, a good picture of the thalamus probably was on the page before that. There's that area, that red area of the brain that we listed there. It's really a relay center. It regulates general arousal and excitability of the cortex. The hypothalamus, which we talked about the 4 F's and finally got a chuckle at everybody that was following along. The hypothalamus, which is on the bottom of page 75, everybody remember this word? What's this word? What's homeostasis?
Internal balance.
Internal balance. Internal, you know, and balancing really your organs, and really, you know, because let's say a person goes into the limbic system activates, and a person goes into fight versus flight. A lion comes through the door in each one of our classrooms. Somebody's had it out for us and has planned a sabotage. And so now we're cornered by lions. And so everybody's, you know, limbic system is going to jump into, it's going to spring into action. It's going to shut down our digestion. It's going to make our heart start beating. It's going to flood our extremities with blood and nutrients. And we're going to be ready to act because we're going to either fight, flee, or freeze. So let's say that now that's, okay, it was a trick. Everybody, they are tame lions. They took them out of the class. Now the hypothalamus, oh, what are you doing? I thought we were going to die. Is concerned with maintaining homeostasis, so getting us back to balance, getting our digestion back online, bringing our heartbeat back under control. Okay, I thought I'd share this, and really it didn't show up at all very well. But has anybody ever seen the movie, I'll reference the movies again, One Flew Over the Cuckoo's Nest? Okay. For those of you that haven't seen it and you're going to be in psychology, it's worth a watch. Who's the guy? I can't remember his name now. He's Jack Nicholson. Jack Nicholson plays a criminal mind who fakes being insane because he wants to avoid his sentence. He wants to avoid going to jail, and so they send him to a mental institution. Well, the bad thing about the mental institution is he gets a taste of an old school, and it's Hollywood, so it's hyped up. But one of the things they did to Mr. Nicholson, and I spoil the ending. It's an old movie if you haven't seen it. One of the things they talk about in the movie, I won't say whether or not they really did this. One of the things they talk about in the movie is doing a prefrontal lobotomy. Now, what is the process of a lobotomy? Let's hear from somebody in Ponca. You guys are conspicuously silent. Let's see. Matthew, you want to take a gander at that? What's a lobotomy?
Isn't that where they remove a section of the brain?
Absolutely.
Correct deviant behavior?
Absolutely, you're right. They would remove, they'd actually do brain surgery, remove parts of the brain to try to correct deviant behavior. So that's what this article is talking about. There in the 40s and early 50s, about 40,000 prefrontal lobotomies were performed in the United States. 40,000. That's, to me, that's quite a startling number. What do we now know? What do we now know? For those of you that studied the brain a little bit, I'll take anybody. What do we now know about the prefrontal cortex? I gave it away just a second ago. Personality. It's personality. You're right. Ben, you're correct. They really, I think, got their information why they were thinking about this. There's a gentleman by the name of Phineas Gage who got stabbed with a spear, some type of accident, but it went through his frontal lobe, railroad spike,
Completely changed.
and it completely changed his behavior. You know, and so they're like, yeah, we can do some good by doing this. What we now know about the prefrontal cortex is this. You know, they're kind of right on it. This is kind of your moral reasoning, your higher level. If you look at a person's brain light up when they're having a religious moment, spirituality, a very deep moment of thought, conceptualizing many things, what lights up is the prefrontal cortex. That's the very last part of you to develop. There are some studies that say our brain continues to add, plentiful neurons up until age 30. It's at least 25. So for those undergrads that are still, you're still under the age of 25, you got some brain growth going on still. So keep it up. Keep on learning. You can pick up new things quicker than the rest of us. So, and some research says 30. But the problem is that when we're thinking about higher level moral reasoning, the best of our best is the prefrontal cortex. If you remove that, I guess the thinking is that they're faulty here. But if you remove that, you remove any ability of us to function on like a higher spiritual Gandhi, Martin Luther King. You think about people that have done things that are very altruistic past themselves. That's kind of the area of the brain that's lit up. That's the area of the brain responsible for it. So they actually, in the end, they really, you'll see, if you ever watch the movie, it's Hollywood, but they're really limiting human potential by doing that. And obviously that's an inhumane practice, and thank goodness it's over. Let's see. Gosh, we took so long to get started. Let's go a little bit further and see if we can get to the action potential. Let's go through the geography of the brain, and then we'll take a little break. Okay.
I have a quick question.
Yes.
You mentioned that the hippocampus had some cortisol involvement.
Okay. Now, what I mentioned quickly there was that cortisol can very negatively impact development of the hippocampus.
Okay.
So I'm just foreshadowing what we're talking about.
Cortisol is the stress hormone. It's produced in times of stress. Could that be why in times of stress we have memory deficits? I'm just wondering if there's a connection.
That's a fantastic question. That's a PTSD question, and I would say yes, because when you have post-traumatic stress disorder, one of the fundamental criteria to have PTSD is to have memory deficit, especially of the traumatic incident. So, good question, and we'll explore that more. I plan to get to a place where we're going to discuss trauma and its impact on our brain. Now, it's not in the syllabus. I plan to get there. Blood-brain barrier. This is an important concept because there's actually a membrane that keeps, and it's a barrier that keeps things in the blood out of the brain. It's kind of like a filter. Okay, think of it that way. There's three possibilities for material that's in your bloodstream. One, it passes easily. So if you think of oxygen and carbon dioxide, those freely pass into your brain from the rest of your body. There are certain things, certain chemicals, certain substances that need help. Amino acids, glucose, and fat-soluble molecules need help in transport. They need, well, we're not going to get into the specifics just because we don't have time. But there are other things that can't pass. They're large molecules, charged particles, ions. You know, they have a strong charge to them. And curiously, water-soluble molecules can't pass the blood-brain barrier. Now, there's a good example, and we'll get to this when we get to our opioids. Chemically, heroin freely passes across the blood-brain barrier. It enters your brain, enters it freely. Morphine does not. There's chemical structures of morphine that keep it out. And so when you have a very more psychological reaction from heroin, hallucinations, dissociation from reality, there's some higher doses of morphine where, you could argue, well, my dad was really out of it when they gave him that morphine. It's not the same. The hallucinations aren't the same. It's much more powerful with heroin versus morphine. So we'll talk about that later on. That's what I can think of as far as pharmaceutically understanding the blood-brain barrier. Okay, nervous system. Now now instead of just having certain areas of the brain that we're talking about, little specific areas of brain geography, we're going to talk about functions. So if we're talking about our nervous system, again, we're going to need to know if we have a central nervous system depressant, we'll need to know kind of what that is. So central nervous system, talking about our brain, forebrain, midbrain, hindbrain, our spinal cord, which is actually, those are, that's part of your brain going down your spine. Our nervous system, our peripheral nervous system, and that's abbreviated PNS. We have a somatic nervous system, so if you think about your skeletal muscles, again, that illustration of moving your hands around, voluntary. Afferent, think about sensory and input, the somatosensory cortex, and efferent, motor and output, movement, that motor, the motor cortex. Now we're talking about peripheral nervous system, autonomic, involuntary. So now let's think more about our limbic system and our hypothalamus. Sympathetic, activate fight versus flight. And so out here we're talking about adrenaline, flowing to our muscles, activating, getting the blood flow up, and and taking the blood out of your, from where your stomach is, and involuntary, parasympathetic, deactivating, bring you back to normal after crisis. So some of the chemical reactions that are going on out here. Peripherally. Does that make sense? Okay. All right. Just to break it down a little more. CNS includes brain and spinal cord, peripheral nervous system, somatic, sensory and motor nerves going to the body and brain. Autonomic, control smooth muscle functions, again like stomach, heart, digestion, and the endocrine glands. Sympathetic kicks in only during fight versus flight versus freeze response, part of anxiety. Parasympathetic maintains calm and steady internal state for most things. All right, so let's watch this video and then we'll take a little break.
The first 4 weeks after conception, neural tissue known as the neural plate forms in the outermost.
So let me preface this a little bit. What you're watching would be the development of a fetus, and so one of the first things that comes online is you think about a new being, what controls us? Our brain. So what should control that new being while it's gestating in utero? Well, it's going to need its neural connections right away. And so brain development starts very, very early.
In conception through birth. In the first 4 weeks after conception, neural tissue known as the neural plate forms in the outermost layer of embryonic cells. The neural plate folds to form the neural groove, then curls to form the neural tube. This tube differentiates into the forebrain, the midbrain, the hindbrain, and the spinal cord. The forebrain will develop into the cerebral cortex, which translates sensory stimulation. It also controls complex behaviors, thoughts, memories, and problem solving. The midbrain will develop into a neural relay station for sending information from the body to various sites in the brain. The hindbrain will control the most basic physiological processes, such as breathing and heart rate. The spinal cord is the pathway for conveying information between the brain and the rest of the body. Between weeks 4 and 8, the embryo grows rapidly and the face becomes recognizably human. The eyes on the side of the head begin to migrate forward and 2 distinct hemispheres of the cerebral cortex emerge. Between weeks 8 and 26 of the fetal stage, the cerebral cortex grows to cover the midbrain. By week 28, a major structural change begins. The cortex expands greatly in surface area and becomes wrinkled and folded inside of the skull. Beginning at week 28 and continuing through week 40, the brain surface fills with hills and valleys, called gyri and sulci. These cerebral folds and wrinkles help to create a vast neural network, which allows the brain to become a highly flexible yet precise control center and processor of information. By week 8, the structures of the brain are already in place, but the amount of brain tissue is rapidly increasing as many new nerve cells are formed. The formation of new cells is known as neurogenesis, or neural migration. During neurogenesis, new nerve cells migrate from the innermost layers of tissue outward. Like gymnasts climbing ropes, the young cells are guided to their final destination by fibers from another type of cell, the glial cell. Glial cells form the underlying structure of the nervous system. Although earlier formed cells rest closer to where they originated, later formed cells move further away from their starting point. In this way, the brain grows, adding layers to itself like the skins of an onion. Once each nerve cell reaches its destination, it begins to acquire characteristics specific to that area of the brain. At this point, each nerve cell sprouts the extensions it needs to communicate with other neurons. There are 2 types of extensions, dendrites and axons. Dendrites look like a network of antennas and receive incoming signals. An axon is a single, larger extension, which carries outgoing signals to other neurons. Synaptogenesis is the process in which synapses proliferate in the brain. A synapse is a tiny gap between the dendrites of one neuron and the axon terminal of the next. It's the point of communication between 2 neurons. So many new axons and dendrites form during the process that it's sometimes called exuberant synaptogenesis. This cell growth process continues into a newborn's first year of life. For an information signal or impulse to move from the axon of one neuron to the dendrites of another, it must cross the synaptic gap. In this animation, an impulse looks like a green ball of light. When an impulse reaches the end of an axon, a chemical called a neurotransmitter is released. In this animation, neurotransmitters look like red balls of light. Neurotransmitters allow an impulse to cross from the axon of one neuron, across the synapse, and onto a receiving dendrite on another neuron. To create a more efficient and rapidly working brain, some of the newly formed synapses are eventually removed. Synaptic pruning occurs during the fetal period, infancy, childhood, and adolescence. It's believed that pruning is a way in which the brain becomes customized to an individual's nervous system as a response to that individual's unique experiences.
Okay, so what struck you about that video?
Mostly that between 4 and 8 weeks, there's a lot of brain growth. It goes from just like the neural tube and the neural tube to actual almost complex org itself.
Yeah, so that neural tube, you think about that, the very basic structures that's controlling that new being is right there from the very beginning. 4 to 8 weeks, like you said. Did anybody get the forebrain, the midbrain, and the hindbrain?
Well, it explains now with like the monster makers how it could actually change the DNA of a child with the drug use. That it explains a lot.
Teratogens. I listened to a specialist on fetal alcohol syndrome, and he said that even more significant than fetal alcohol syndrome, because fetal alcohol syndrome was talking mainly about the physical effects of the flattened, what's that called right here, Ben? Well, it's a flattened palate, and you have the eyes that are farther apart. And there's very striking features about it. Some children that have fetal alcohol syndrome. What he spoke about was fetal alcohol effects, because those structures come online universally between. He talked about there being specific days in development, like between 21 and 23 days of the second trimester. I mean, basically, the guy went into, we're very, very similar when we get on that very basic genetic level, that our development happens between 4 and 8 weeks. And he could basically tell by a physical feature, like if a child was born without a couple of fingers, he could say, well, that was on this particular date in development in utero, the mom drank heavily. And so what he talked about were fetal alcohol effects, and specifically too, he said a lot of times a child may have fetal alcohol effects, but not be detectable physically. You won't be able to see it in outward appearance. But what he has done is he's actually done some, and it's very expensive to do MRIs, but he's done some MRIs on some children that he suspected to have fetal alcohol effects, and is able to look at how the corpus callosum, how it's developed. If it's slanted a certain way, you're talking about internal brain structures. The person looked normal on the outside, but because of particular days when the mother had ingested, different chemicals, it had affected and really caused some maladaptation in that early development. So now, we hear we're really sensitive on caffeine. Well, the mother shouldn't have so much caffeine. Well, caffeine shuts down, the blood flow. It's a stimulant, and it shuts down the blood flow to the uterus. We'll get into some interesting things there. Also, if you're talking about maybe even methamphetamine or cocaine use, those substances actually induce pregnancy. They induce contractions in the smooth muscles. So they actually can, you talk about premature birth, low birth weight. You can actually have a premature birth induced by, a massive ingesting of a stimulant. So, we're to a really profound point in our science where we're able to really narrow down what's happening. And so, good question. You kind of got me off on a rant a little bit there. No, that's good. It was good. Did I answer your question? Okay. Thank you. Let's take anything else on that? Okay. The last thing, the front, the mid, and the hindbrain. The midbrain that they're talking about there has a lot to do, like the hypothalamus, the limbic system, some of those things. It's kind of a switchboard. It's like, okay, what do we do with this information and transforming it? Does it need to be action? Does it need to be like increased heart rate? Do I need to feel an emotion with this? I mean, it's just that midbrain is not quite thinking. It's still a little bit it's kind of in the transitional stage between thinking and automatic processes. So I didn't really go into that too much because I wanted to simplify automatic versus voluntary. Okay, let's take probably 5 or 10 minutes, take a break, come back and talk about action potentials. I'm just going to step out of here because it's so hot.
I just do. Because
So Bailey, how far did they move you all there in Alva? Well, I think I'm on mute. Yep. So Bailey, how far did they move you all in Alva?
They didn't move us very far, just over to another building.
Yeah, they moved you out of the building even?
Yeah.
Oh my. Well, sorry about that. I don't think that I kept trying to dial y'all and it wouldn't even go through. So I don't know.
Yeah, we didn't know what was wrong with the system in there.
Thanks for waiting on me. I'm not a PhD. Y'all get extra credit for waiting the extra 20 minutes. Oops. Oh. Get this back. All right, well, let's go ahead and keep on going. A little bit of follow-up on the neural development of the child, just so that you have of the fetus developing human. That's a crash course. You can do your own semester in fetal development, but you see a timeline at the bottom. Really on that next slide, it gets a little bit more where you can see it. And so you kind of get an idea of what comes online and when, how the developing person's brain is happening. Brain density, synapses, greatest at age 6. So that fourth bullet point down. Actually, you see that neural migration takes 12 weeks. The brain makes 150,000 neurons per minute. 150,000 neurons per minute. That's making some brain cells. And there are maybe 1 trillion in the brain at 12 weeks fetal growth. So your brain does a lot of morphing early on. You actually have more brain cells. You're born with 1 trillion brain cells and neurons, and that comes down to 100 billion. So, they postulate that in that first year of life, you're losing a lot because your brain is adapting to its environment. It's becoming online. You're becoming human, and you see synapses greatest at age 6. You have some neural pruning that takes place that they talked about a little bit in that video. You're like, pruning's bad. We talked about that with meth last week. If you lose connections, you're losing brain power. Well, the science here, when you're this age, the pruning goes to specialize you for whatever your environment is that you're growing up in. In other words, we have a lot of versatility. We have a lot of different directions we can go. Early on, a human, when I was in school, they said there's a babbling stage, and humans babble the language that they're around. That's actually incorrect. Humans are able to kind of babble syllables, phonemes from all languages when they're very, very little. But as we grow and we hear languages and they're spoken around us, that becomes very specialized. So, for instance, you hear someone that speaks an Asian language, normally they have a hard time saying the ahs and the l's, because in their language, those sounds aren't really, they're not prevalent, they're not pronounced. And so, in our, if we have a hard time like learning in Spanish where they roll their R's, like, well, let's see, what would be a good, rojo. If you're going to say rojo, now I sounded like I was from Oklahoma. They were saying rojo, which means red. My little 2-year-old's learning to sing some song about red is rojo, and so I try to give him the little R roll there. Some folks really can't do that. And it's just because that's not a sound that's made in our language. So you can kind of see where our brains might lose some connections because we're specializing ourselves to the environments in which we grow. You'll notice at the very a couple things in the bottom. Well, let's see. Important for our purposes here, third from the bottom. All psychotropic drugs interfere with neural migration in the fetus or otherwise influence brain development. So I have an expectant mother that I'm treating, and her, I'm going to leave the pharmaceutical dosing and prescribing to the doctors here. They took her off of Zoloft and curiously left her on gabapentin, which to me is a more potent drug. It's, at least from my understanding of it. I don't know if it's had the research on it. That being help me with the help. That gabapentin. What's the
drug classification?
no, the drug classification.
I'm not sure, but I mean,
It's I have it. I can't think of it.
Is it not like a nerve blocker?
It is a nerve blocker. It is a nerve blocker, and it seems like it's a more potent. But they left her on that.
The drug book will tell. That's what they look at.
Yeah.
There's like a category in there.
Like not for pregnant women.
Right.
Pregnant women can take. So I'm a little skeptical on it, but we'll see. Gabapentin is the drug. I'm trying to think of the common name for it. Neurontin. Thank you. Yes. Yeah. Thank you. Yeah.
You're skeptical of gabapentin?
I'm skeptical of giving it to a pregnant woman.
Oh, okay.
Because to me, it's a more potent.
Wouldn't that depend upon what it reacts to in the woman's body? I mean, whether it would actually, how it would affect the fetus. Gabapentin and Zoloft would affect two different areas, wouldn't it?
And what were you gonna say, Ben? They passed the placental barrier. Yes, thank you. Thank you. So, but to me, there's still chemicals. There's got to be something, with. I appreciate that perspective. The placental barrier does protect the fetus from some of what is in the mother's body. But again, I'm still. The jury's still out there. We don't know. To me, we still, although we've got all these advances, still don't know enough about all of human life development. Our cells on a very basic genetic level. So we'll see. I mean, I'm if he's he obviously he's practicing medicine, so he feels comfortable and I'm just saying from my own experience. I'm not saying as a medical expert, because that's not my expertise. Axonic migration continues through most of our lifespan. So we can learn, you can teach an old dog new tricks. Our brain continues to develop and change until we die. The hippocampus is one area of the human brain that is known to generate new cells. So our memories. And here's kind of a table for development there. Okay. Let's move on. Basic structure of the nervous system. We've already identified. We're pretty comfortable with the 100 billion neurons. That number of brain cells. They do specific things. Now, when we're trying to define them verbally, they analyze and transmit information. So we learned before, like the nucleus in each one of those neurons makes decisions. do I send that impulse on or do I stop it? You know, is it important? We'll talk about what that looks like in an action potential in a few minutes. We receive they receive sensory information from outside the body. So how hot is it in Enid? I detect it's around 82 degrees in room 221 here in the ITV studio. I can feel the sweat on the back of my neck. I'm receiving that sensory information. Those of my Enid colleagues are wrong with me. Integrate and store information. So again, our hippocampus and integrating, making sense of the information around us. Controlling the muscles and glands. Basically responsible for everything we see and understand as behavior. Those are your neurons doing that. They're the main players in the orchestra. Neurons come in a variety of shapes and sizes, but all have some basic characteristics. So I showed those pictures. We'll get back to those in just a minute. The dendrites. And what do the dendrites do? Alva, what do the dendrites do? Seth, what do the dendrites do, sir? Okay, if they're playing in the Pro Bowl, they're wearing number 80 and they're running down the field.
They receive the information.
Yes, they're getting ready. They're receiving the information. Absolutely, they're receiving projections. Though that's an important thing. That'll be testable material. Cell body in the soma. I call it the soma. It's really the cell body. It's got cell wall surrounding the nucleus. Contains the nucleus and genetic material. And then thirdly, we have an axon. Now again, let's see here. Rachel, what does the axon do?
It sends the information.
Thank you. Yeah, axon is sending the information. Send that up. So think about that lightning bolt from last week, shooting down. That's the axon. Connects to other cells and transmits signals. Now, in just a minute, we're going to talk about other things that are called glial cells. We didn't talk about those last week. These things are influenced by glial cells, myelin sheath, and they create nodes of Ranvier. So here's our illustration from last week, our soma, our nucleus, our dendrites, which are receiving information, our axon, which is sending information. Just another illustration of it up there. The neurons, to me, are the CEOs. They're the ones that are the decision makers. They're the ones making things happen. Not that the other people in the company aren't important. You have three basic types of neurons. You have sensory neurons, that somatosensory cortex we talked about, receiving information through the senses, touch, pain, vision, hearing, taste. The occipital lobe back here is taking the visual information in. Sensory neuron. You have motor neurons, controlling muscles and secretions. I have some motor neurons, I guess. It's helping secrete some sweat, like back here on the back of my neck right now. It's actually helping me move around. It's helping you move around. And then we have interneurons. The interneurons are the most numerous in the brain. Interneurons basically communicate and connect with each other. So we're just talking about brain cells in here that are connecting and communicating amongst each other, not sending information out here or receiving information from the outside world. A majority of neurons are interneurons. Important to remember. More information about neurons. Talking about the axon. An axon can be as small as a few millimeters up to a meter in length. So think about an axon that's got to run down your spinal cord to control your leg. That's got to be a long axon. Some neurons originate in the brain and have axons that extend into the spinal cord. Many axons are coated with a fatty substance that is actually a glial cell. So we're going to talk about glial cells next. They're very important. which wraps around the axon. Nodes of Ranvier allow high-speed neural transmission of up to 200 meters per second. That's some fast movement right there. This myelin sheathing is not completed for several years after birth and appears to continue to up until late teens. And I added this, that was the one I inherited. Myelin sheathing, development of myelin continues into adulthood too. I mean, you can teach a new dog, or an old dog, new tricks. Sorry, but dyslexia is kicking in there. So now let's move into glial cells. Let's understand glial cells a little bit. Anybody have any questions on this last slide here? We're gonna start to understand what this means. Glial cells are the support crew. Now we already said how many neurons are in the brain? About 100 billion. There are equally, roughly, the same amount of glial cells in your brain. They're roughly 100 billion as well. They're just as numerous. And there are several different kinds. So we're going to try to understand what those. Here's the different kinds. We have astrocytes. Astrocytes supply the neuron with nutrients. They're star-shaped. They basically say, hey, you know what? We need to supply, this neuron needs to live and breathe and act, and so we're gonna get some nutrients for this neuron. Microglia, and glial is a fancy word that means glue. They thought, when they first looked at the brain and they saw these other cells around the neurons, they're like, hey, that's the glue that's holding them together. So they called them glue cells, glial cells. They're not actually, they've got a lot. Pardon? Jennifer, did you say something?
No.
Okay, sorry. I heard something come over from a remote site, and you showed up, so I thought maybe you said it.
I dropped my pen on the desk.
Okay. So keep everybody awake there in Woodward, all right? I'm joking with you. Thank you. Microglia are responsible for cleaning up and removing waste. So those are they have some specialized functions there, feeding and cleaning up. Then you have an oligodendrocyte. Now these are myelin sheaths in the brain with your central nervous system. If you have a myelin sheath, we'll see in just a minute, it makes a dramatic impact on how fast information can travel in your brain. It makes a really dramatic impact. Schwann cell, and we'll see a Schwann cell in action. These are myelin sheaths in the body, so out here. So right now, you guys notice I move my hands a lot. They're well myelinated because I use them a lot, and so the more that you use something, the more, generally speaking, the more myelin that you generate to help you reinforce what you're doing. This is an exciting part of learning because glial cells have a lot to do with helping you learn. Not just make new connections in the brain, but reinforce the ones that you have. Myelin sheaths in the body, peripheral nervous system, motor neurons. So they basically, these two basically mean the same things. They're making myelin sheaths. These are in the brain, the oligodendrocytes. Schwann cells are out here in the body. That's important. So how do they interact? Let's check this out.
What you will see and hear in this presentation is the development of myelin in the peripheral nervous system and the propagation of the action potential along a myelinated axon. This multimedia presentation would be most helpful if you already have a good understanding of the Schwann cell and the electrochemical process of the neuron, called the action potential. The Schwann cell forms a protective covering around the axon. Schwann cells start to develop in the embryo and continue to increase the wrapping around the axon through childhood. This development increases the thickness of the wrappings, which peaks in adolescence. This is why teenagers have such quick responses. The Schwann cell contains the typical cell organelles and cell membrane structure. However, notice as the Schwann cell surrounds the axon that the nucleus and other organelles are squeezed to the outside wrapping of the cell. This outer wrapping of the Schwann cell is called the neurolemma. The inner lining is made up of layers upon layers of cell membrane. This inner wrapping is called the myelin sheath. You will recall that the cell membrane, called the fluid mosaic model, is made up of a bilayer of lipids integrated with proteins. The thicker the myelin, in other words, the more layers of cell membrane making up the myelin, the more advantageous it is to the axon. One advantage is the regeneration of severed axons. Another advantage is an increase in the speed of the propagation of the action potential along the axon. The rest of this presentation will concentrate on the increased speed of action potentials down the length of the myelinated axon. Here is the neuron, and you can see the repeated Schwann cell membrane forming the myelin. Note that there is a small space between the Schwann cells where the axon is not covered by the neuroglial cell. These spaces are called nodes of Rampier. From what you already know, action potentials occur at the axon hillock and continue to be repeated away from the cell body, much like dominoes falling one after another. An action potential starts on a polarized membrane, which is -70. A stimulus causes the sodium gates to open slightly, and sodium starts to trickle into the cell. If the cell reaches -60 or threshold, the sodium gates open wide and sodium floods in, bringing the inside of the axon to +30. At this point, the sodium gates close and potassium gates open. Potassium starts to pour out of the cell. This allows the neuron to become polarized again. Then the sodium-potassium pump starts to actively transport sodium out and potassium back into the neuron. First, we will look at the propagation of the action potential in the unmyelinated axon. Propagation is the repeating of action potentials down the axon. The action potential is repeated because as the sodium comes in, it diffuses to adjacent areas within the axon. As the sodium increases in this area, threshold is reached. Sodium gates open wide, sodium rushes in, causing depolarization and an action potential. As the sodium enters this area, it diffuses through the axoplasm and another action potential is created. This continues down the length of the axon. Now look at the myelinated axon. The same process applies to the myelinated axon. An action potential develops, and as the sodium comes in, it diffuses through the cytoplasm of the axon. It continues to diffuse through the portion of the axon wrapped in myelin. The increased sodium concentration reaches the node of Rampier, increases the axoplasm to -60, and depolarization occurs. The sodium gates open wide, sodium floods in, and we have an action potential. Again, the sodiums diffuse through the axoplasm, reaching the next node. An action potential develops. The process is continued down the myelinated axon, passing from node to node. Compare the unmyelinated axon with the myelinated axon. You can see that action potential reached the end of the myelinated axon more rapidly than the unmyelinated axon. The speed of the propagation is faster going from node to node than action potentials that develop adjacent to the previous action potential.
Okay, so a lot of that was jargon, I know, and we'll get into a little bit of that, but the main thing that I want you to recognize from this is this last little bit, this 420 moving forward.
Compare the unmyelinated axon with the myelinated axon.
So which one is moving faster? The myelinated. So what's happening is you're having the impulse, the action potential started from this nucleus. You know, this cell received information from other axons, from other neurons, and decided, you know what, that's important. I need to send that on. And so this is illustrating a connection that is probably used quite a bit in this person's brain, because there's a lot of myelin, there's a lot of these glial cells that have formed along this axon. And so what happens is they bunch up and they create little gaps, and those are called the nodes of Ranvier. Okay? And what happens is the potentials just kind of skip along this, The sodium potassium pumps, well, we'll get into what those are, but they skip rapidly along this line. Versus, and this when I first learned this, I was like, that doesn't make sense, but you know, if you think about the way that it condenses the action here, it does reverses the unmyelinated axon, which this is taking forever. I mean, this is, I thought this was the best illustration I could find.
Reach the end of the myelinated axon more rapidly than the unmyelinated axon. The speed of the propagation is faster going from node to node than action potentials that develop adjacent to the previous action potential.
We used to use an analogy that you had one person running, trying to get to the end. You had other people trying to pass 3 mountains, and you had people smoke signals on each of the mountains. Once they saw the signal, it's who's going to get the message to the end faster, the runner or the people with smoke signals.
Pretty good analogy. I'm with Ben on that. You do have a lot faster communication. To break that, to boil that down, let's go in a little bit deeper, and then we'll boil that down a little bit better. I know, don't get too hung up on. I'm in disagreement with some of her numbers on -60, but we'll get into that in just a minute. I did think that video had the best illustration of the quickness that's added with myelinization with the glial cells. So, coming back to let's land for a minute. Neurons are the brains of the operation. Glial cells do the busy work. They offer protection. You said, if an axon is severed, they act as a way to help reconnect those axons, to repair them. They supply nutrients and oxygen, remove waste, destroy dead neurons. So, if you think about that little baby's brain developing when they're having, this cell death, which sounds horrible, but it's the baby reacting to its environment and getting used to it. Glial cells are doing a lot of this busy work and helping reformulate the brain. They significantly, for our purposes here, significantly speed up communication through myelinization, through oligodendrocytes and Schwann cells. And this is fascinating. This will kind of help us understand myelinization, because who had the sleep and dreaming class? Okay, so we had a couple people take the sleep and dreaming class. Did they talk about REM sleep aiding in learning? Okay, so we're going to talk about that on a very basic level right here. That's going to bring us home on why is myelinization important, and why is sleep important as well. So we need 340 to 820. Let me go to 340 here. Whoa. There. So give this just a moment to load.
I have a question.
Yes. Let me make sure this is going to load for us. Okay, yes.
Is there, see how the axon, why aren't all axons myelinated? Is it just the myelinated axon? Is just like repeated use of information? Wrapping of
it a little bit depends on our neuroplasticity, our age, on the area of the brain that we're talking about. It depends on usage. Let me show this video, and I think it'll go to some of your question there.
Okay.
Because you make a logistic point, a logical point is, why don't they all have?
Why is it important? But it would be an
right.
overload, wouldn't it? If all of the axons.
That's a great question. I assume it would be related to an efficiency of energy and making sure that the energy in the brain is going to where it needs to go. Let's see here. There was Brittany. You had made a comment about using 10% of our brain, right? Okay. Who's ever heard you use 10% of your brain?
Yes.
I heard that going up too. Actually, that is a complete falsehood. We use our brains. I mean, you can use the PET technology, the MRIs. Our brains, we use our brains daily. Most of our brains. I mean, if you don't use a part of your brain, kind of where you're going through, it's gonna atrophy. It's gonna wither. It's gonna die out because our bodies are efficient with same way with our muscles. If we don't use them, we kind of lose them. So think of it kind of that way. Think of it as being like, okay, well, I haven't studied. Like I haven't studied algebra in years. And so if I'm going to go sit down and try to pass a GRE algebra section, that's not going to be good because that area of my brain is not myelinated right now. I might study some different aspects and think about, okay, well now that's kind of coming back to me, and you got to divide right here, and here's the operation, and here's the step. But unless we use that, that's wasted. And so the 10%, we're really good about maximizing the energy that's available to us. And so that's just not, the 10% thing is absolutely not true. Now, categorically, I'll say that is not true.
Have you seen that new movie Lucy that's out?
That's what I was talking about. That's what I brought that up.
I have not seen Lucy.
Yeah.
It's interesting.
It'll leave you silent.
Yeah, it's interesting to think about.
All right, I found this finally loaded, so we'll listen to some of this.
Perform and react appropriately.
Here on again.
Professor Carlisle Smith at Trent University in Canada specializes in sleep and learning. We asked Dr. Smith to run both Nicole and Charlie through a series of sleep studies. Both teens will perform certain tasks, go to sleep for the night, and come back 3 days later to repeat them. It doesn't matter what skills they have coming into the test. It will simply measure under various amounts of sleep whether they will improve their skills.
15:47.
Almost half the time.
You're on there half the time. That's pretty good.
The exercises are designed to get the brain learning something new.
Stop. See what you got here? Look at Charlie on his first trial. There, just a minute.
This test is a real brain teaser. It's like learning an equation in math. Not only does Charlie have to trace something upside down, he must visualize the task in his brain and work from that picture.
Can't get out of this. The special worry with teenagers is that they are learning a tremendous amount and trying to keep up with their peers and so on. They're often stretching almost to the limit of what they can do. And sleeping in is one of the best ways that they can do to sort of stay abreast of what's going on and let them do it.
Yeah, who makes the beds?
I do.
Can you make mine again?
Yeah, I'll make it up for you. Did you mess it up?
No, it was just from everybody being in it.
Yeah, I'll make it nice for you.
Hospital corners?
No.
No?
No, I'll give you lab corners.
It would seem hard to go to sleep this way, but Jeremy Jacob needs to measure exactly what stage of sleep Nikki is in. He's particularly interested in REM, rapid eye movement, a time of dreaming and learning. The same part of the brain that was working when the teens were learning their new skills continues to rehearse and practice when the students sleep. The brain consolidates and improves on what they have just learned. In a sense, the lessons are effortless, happening while they sleep.
The best predictor of how well someone is going to do, be they at Harvard or wherever, is not their SAT scores or anything else. It's whether or not they get a good night's sleep. And then up again to 2. And then
3 days later, the teens are back.
Charlie and Nicole, one of you got a normal night of sleep almost. What normally happens is you start off with what's called a little stage 1, and you go to 2, 3, 4. Nicole, she got 105 minutes of REM sleep. That's quite a bit. Charlie, on the other hand, you only got 44 minutes. You got less than half of what she got. So you're a little short on REM sleep, and as it turns out, that's going to be pretty important as to what happened to your tests.
Smith had purposely arranged for Charlie to have less sleep. Now, after retesting the 2 students, the evidence is clear about how important sleep is to learning.
You improved by 6% on the ball and cup.
I improved?
You improved by 6%.
I thought I did crappy.
No, you improved by 6%. But Nicole improved 11%. We're looking at your degree of improvement. And in terms of improvement, you didn't improve as much as Nicole did. So she was over double what you had. Now then, the mirror trace. This, we think, requires REM sleep. Nicole, who got lots of REM sleep, she improved 44%. And you, Charlie, you were worse by 10%. I can't tell you any more clearly having a good night's sleep will give you more of an advantage than anything else you can do. Now, you can relearn this stuff, but you're going to run out of time. Every day you say, well, I'll catch up. But while you're catching up, Nicole is busy going ahead. I think maybe you should go to bed now.
In response to the studies that show many teenagers are sleep deprived, a number of school districts across the country have changed their school start times.
Oh, I don't want to get into that. That's another subject. The main thing that I would like for us to note there is that at one point they said the same areas of the brain are lit up, and while they're sleeping, that were lit up while they were doing those learning tasks. And so this study was done in the early 2000s. Now they're getting to where they can see, your brain actively myelinating, the connections, the axons. And so when you get REM sleep, this is important for y'all as you're still studying. When you get a good night's sleep, your brain is reinforcing your learning that took place that day. Your brain is actively changing. And so even as adults, we're still learning. We're still getting that myelinization. So if you go home and you study this and you journal it and you recall some of it, your brain's creating some myelin. And so when you come back and you test, it'll be that much more reinforced. So good stuff there. Any questions or thoughts? Okay. Now the tricky stuff. Now that was all the fun. That was all the fun applied science. Action potentials. Now you heard that lady at the beginning of the video with we're talking about sodium, we're talking about potassium, where it's coming out. They had the different numbers on there. Neural transmission is actually a 2-event phenomenon. The first type of transmission is electrical. We know that. Neurons, the brain cells, neurons typically have a resting potential or charge difference of -70 millivolts. So your neurons are their natural state is negatively charged. The outside of the cell is positively charged compared to the inside of the cell. Large protein molecules are embedded in the cell walls, which serve to pump ions out and maintain the resting potential until the cell fires. So it's going to let sodium in just a moment, and we're going to have depolarization. We'll talk about what that is. These and other large proteins at other sites, such as the synapses and dendrites, are often the target sites for psychoactive drugs. That's why this is important. All right, so here's an action potential illustration. Uh-oh, we are really quiet. Oh, I got really quiet. Can anyone hear that?
Hit it with a hammer.
I'm ready to tonight. This has been challenging. Basically what it's showing, if it were at all audible, it's showing, here's your axon membrane. Okay? You know, it's a little tube that's going to send your electricity. Naturally, the inside of this is -70. The girl said it was -60. I disagree. I think it's -70. That's the sources that I have were pretty conclusive until I watched that video. When an action potential happens, there's a sodium and potassium exchange. So you remember your from science class, from chemistry, your periodical chart of the elements. Sodium being Na, potassium being K. You know, positive charges here. These are your sodium potassium pumps, the large protein molecules. They're trying to illustrate, there's the resting potential, -70 millivolts. That is called polarized, and it's using the British way that you spell polarized. Your resting potential for your neurons is polarized. It's -70 millivolts. It's not neutral. It's not a neutral charge. It's negatively charged. So then we have our illustration of our dendrites and our nucleus and our cell body, everything that we've already covered. Our axon hillock, which is the beginning of the axon. So now we have a lot of potassium that's naturally, am I getting that right, Ben, or I have it backwards? Potassium is in the cell in the resting potential, yeah. Potassium is in the cell. We have a lot of sodium outside of the cell before the electricity happens. Okay? So the impulse starts, and this is what gets the impulse going, is all of a sudden these gates open. These gates open and sodium comes rushing into the axon. And so this is what's happened on that microscopic level at the nodes of Ranvier.
You said sodium is inside the cell at resting potential?
Resting potential, it's potassium.
No, that's inside the cell?
That's inside the cell. So there's an exchange. Basically, let me see if I can write this down. So, now I wish this was a little bit better illustrated. Let me keep going and see if they give us something better to write about here. Okay. So here's your electrical current. Something happened. A neuron, you know, the nucleus of a neuron said, this is important. I need to send that information on because I'm no longer at my resting potential because it's this chain reaction that keeps on going, keeps on going. I got all this information from my dendrite and it says, yeah, Taylor's talking. Let's send information about Taylor because he's the most exciting thing in the universe talking about neurotransmission right now. So what happened then was this -70 millivolts depolarized. And now you're actually going to go up to about a positive 40 millivolts. And your sodium ions diffuse in. Sodium comes rushing in to the axon. And that pushes the electrical signal down the axon to the end of the axon terminal. And then that releases the neurotransmitters. You following me? Okay. Cool. I'm glad I'm funny to somebody somewhere. Okay. because this is probably the dullest part of the illustration. And so basically,
in the hallway.
Oh, it's in the hall. Okay, I wasn't taking offense to anybody. I figured it was crosstalk. Okay, so then this is another important point where this guy's talking. So it reaches about plus 40 millivolts. At that point, we're like, we've got enough sodium. No more sodium. The sodium channel is closed, and the potassium channel is open. So it's a sodium-potassium pump. The sodium now pumps out, and the potassium pumps back in. Potassium ions diffuse out, and potassium ions diffuse out. So we pump out potassium too. Okay. Sorry, I gave you some wrong information there. Follow this, because I do trust this source. I wish I could hear what he was saying. This is by diffusion. I still say the potassium. I still say the potassium keeps us level. I think Ben was correct in that. There's a little bit of a hyperpolarization and diffusion through the membrane. And there's something called the refractory period. And we'll talk about what that is. All right, so I apologize for the confusion there. I planned on that being audible. Action potential. When the cell is stimulated at the dendrite end, it allows an influx of positively charged ions through the holes in the cell wall called ionophores, or ion channels. This depolarization, so when you have an action potential, you're depolarizing because you're naturally polarized. Proceeds in a domino effect fashion from the axon hillock to the ends of the axon. The axon obviously ends with a synapse. Okay, so let's talk about this this refractory period. So you get, let's say there's little bitty stimuli, kind of like this sign over here. We used that last week. There's little bitty stimuli that you guys hadn't noticed the sign until I just said that. You guys were getting a little bit of input, a little bit of visual stimulation. Your dendrites were picking that up somewhere in your visual pathways. But it wasn't really causing action potentials in the part of your brain that's controlling your attention. The minute that I pointed to the sign and I said, okay, there's a sign there, boom, you had an action potential. You had depolarization in the parts of your brain that control your attention and your vision, and the motor neurons that control where your eyes are looking. It met the negative 55 threshold. It was like, oh, that's there now. I can process that. Boom! And it started this chain reaction that helps you process that sign. Now, when you turn away, when you stop thinking about the sign, because you're trying to pay attention to the lecture too. You can't just, wow, that sign is so pretty. There's white here, and there's black letters there, and there's little red letters, and it's kind of the fan's making it move. You can process a lot about that sign, but your brain moves along relatively quickly. You have a refractory period, so you just can't keep going. And the guy talked about your cell has to rest sometime. It's kind of like if we just had every cell flipped on the whole time, we'd never attend to anything because we'd be flooded. And I have a good way that I remember this in undergraduate school. What does this have in common with male orgasm?
There's a period of you can't go back into it.
There's a period where you just can't go back into it. Thank you. That's a perfect. I appreciate that. Brittany, for a $64,000 winner for having the courage to say that. Just our cells are just like male orgasm in that they just can't stay on all the time. There's a period where you just can't go back into it. That is the refractory period. So if you're thinking about that and you're trying to recall a refractory period for the exam, oh, I just had this wonderful experience. Okay, I'm gonna have to wait a little while to have another one. So that is refractory period. Any questions on refractory period? Ponky, you look concerned there. Sabrina, you got any questions on refractory period? Nope. Okay, just making sure. All right. I show a lot of videos because I want this to, I want this to come alive for you. I want this to make sense. I could talk about sodium potassium pumps and I will send out a clarification now because I've muddled that the potassium and the I still say there's potassium in our arresting potential, but at least you can kind of hear this one. I don't know why that other one was so quiet.
Oh, only you done me and you bet I felt it. I tried to reach you, but you're so hot that I melted. I fell right through the cracks, and now I'm trying to get back. Before the cool done run out, I'll be giving it my bestest. Work it, make it, do it, makes us. Harder, better, faster, stronger.
Okay, I'm going to interrupt Daft Punk for just a minute. You'll notice it said that neurotransmitters impact neurotransmission by exciting or inhibiting them. So we're going to talk about that in just a second. We've talked about excitatory stimulation so far. It's important for us to get inhibitory because when we talk about GABA, GABA inhibits transmission. Sorry, now enjoy the music. All right. Thanks for putting up with the music there. Try to keep people awake at this point in the presentation. Any thoughts on that? Just another look at it. I mean, it's just if the more that you get a look at, the more you think about it, the more it starts to make sense. Dopamine system. If this one's not too long, we'll watch this. I think this is only about a minute long.
Our brains are complex webs of chemicals and nerves. Inside the brain, nerve cells called neurons are constantly communicating with each other to shape how we think, feel, and act. Let's have a closer look at these nerve cells. These are the ends of two neurons. The one on the right sends a message, the one on the left receives it. At first they look connected, but they're actually separated by a tiny space called the synapse. This is where messages are relayed. What we'll see next is how we normally experience pleasure. The sending neuron contains dopamine, the brain's pleasure chemical. When something good happens to us, this feel-good chemical is released into the synapse, where it connects with receptors. There, dopamine activates the receiving neuron, which in turn conveys the message onto the next neuron, creating a chain reaction that produces pleasure. After the message is sent, dopamine is recycled by transporters to be reused. This conversation repeats itself again and again, and gives us the feeling of pleasure and reward.
Okay, again, eye on the prize. That simplifies it, kind of like I did in last week's class. We will be referring often to the neuron, and that's, again, I'm not going to take responsibility for that, in two ways. And that's supposed to be the number two, presynaptic and postsynaptic. Presynaptic, the axon; postsynaptic, the dendrite. As we discuss functions of the axons, presynaptic, and dendrites, postsynaptic. Stimulations of the dendrites and cell body. The axon has only voltage-gated ion channels and doesn't really have much variety of response. But the dendrites and cell body contain large numbers of protein molecules and genetic material, which can cause a variety of cell responses to stimulation. We refer to the depolarization of the dendrites and cell body as postsynaptic potentials in the dendrites, PCSPs, which are converted into action potentials at the axon hillock, where the axon joins the cell body of the cell that's receiving the information. Synapse, or synapse, as the lady said on, sorry, synapse for our purposes. The way the nerve cells communicate at the synapse is the very heart of psychopharmacological interest. This is because drugs affect these synapses. In fact, the neural transmission is mediated by a group of chemicals the cells manufactures, which are known as neurotransmitters, which we've gone into. There are at least 50 well-known neurotransmitters, but there are likely a large number of other ones yet unknown from your text. Key terms are highlighted. When the action potential reaches the ends of the axon, it causes the release of neurotransmitters into the synaptic cleft, or gap, or synapse, between the two cells. These neurotransmitters are actually stored in vesicles. So in the axon, before they go out, you saw the Daft Punk They were coming down in the vesicles before they hit the synapse. Vesicles store the neurotransmitters, which are moved to the edge of the cell membrane when it depolarizes, and then they release their neurotransmitter. As a rule, each cell releases only one type of neurotransmitter at all times. I believe that's called Dale's law, but I don't know who Dale is. Synapses. The neurotransmitter travels across the gap and lands on receptor sites on the postsynaptic cell. These can actually be on the dendrites or the cell body wall itself. So I've really been focused in on the dendrites. Axons can actually, and the cell body can receive some information as well. Receptor sites have protein molecules embedded in them which are specific to the neurotransmitter. When the neurotransmitter lands there, this is when I stopped the music, the record scratched and you heard, whoa, wait, this is important. It can either reduce the postsynaptic potential, bringing it closer to the firing, or increase the postsynaptic potential, making it less ready to fire. So it could hyperpolarize. Instead, it rests. You have some of them where your natural neurons are resting at -70. Sometimes you have some chemicals released that push it down to -75 or -80. The trigger for action potential to me is if that, from what I've learned over the years, instead of -60, is -55. So that's the number I want us to know for this class. If a neuron can be triggered to depolarize to -55 millivolts, an action potential will ensue, and it'll send the message. Some neurotransmitters are excitatory, while others are inhibitory. They inhibit neurotransmission. They inhibit action potentials. Fast-acting transmission, when a receptor site is stimulated, it can result in changes in the ion levels in the cell, which makes the cell more or less ready to fire, as stated on the last slide. This is either fast-acting excitatory inhibitory function of the neurotransmitter, but there is a slow action process. The slow, long-acting transmission, this has to do with some of the different chemicals we'll be looking at over the semester. Again, it will get interesting because we'll see how these drugs really do this. Secondary messengers. Some neurotransmitters and drugs influence the presynaptic cell, postsynaptic cell, by causing the cell to release a special molecule inside the postsynaptic cell, which can either influence ion channels from the inside, or they can travel to the cell body and cause certain genomes to become activated. This is getting technical. Don't worry about memorizing this. We'll get there. Which can alter the receptor sites, ion channels, and other cell functions, including apoptosis, cells committing hurricaria. That's from Dr. Davis. I didn't put that in there. At every point in this process, drugs can influence and alter the outcome. So there you see, this is one of Dr. Davis' slides, apoptosis, where a cell can actually decide that it needs to die, or necrosis, where something else kills the cell, which a lot of chemicals, alcohol being one of those, can cause, necrosis. Slow, long-acting. The process involving secondary messages may take minutes to hours to days, and may have effects that cascade down for even longer and can alter to receptor sites more or less permanently. In fact, it is now known that the receptor site can even talk back to the presynaptic site and influence the levels of neurotransmitter being released. The second messengers also play a role in upregulation and downregulation. Changes in the number of receptor sites. We know that when we give a person an antidepressant such as Zoloft, yeah, it's making your serotonin work more. Remember, it blocks the reuptake, so that the serotonin's out there in the synapse longer. But what happens is your cells tries to adjust, and actually the postsynaptic cell will start to reduce the number over time of serotonin sites of its ability to take on the serotonin because it's had more. And so that's why it's important to, if you're taking Zoloft or Paxil or Prozac and SSRI, to give it a month or so before you really recognize what it's doing in a person's body. That'll be important. This talks a little bit about secondary messengers. We're getting late, so I'm going to keep going. Terminating synaptic action. after a neurotransmitter is released into the synaptic gap, it travels back to the receptor site. Afterward, it's released back into the gap where it may either be metabolized. You know, the enzymes can come and eat it. You know, some of the glial cells may take it, or it may return to the presynaptic site and be placed back into storage vesicles for later use, reuptake, the vacuum. Again, protein molecules transport the neurotransmitter across the cell wall and into storage, reuptake. Drugs may alter reuptake, we watch that, by blocking uptake channels in a number of ways. Autoreceptors may also increase or reduce the amount of neurotransmitter being released. Neurotransmitters, we went over these. We talked about 4 of those. Another one we'll talk about is acetylcholine, involved in involved in muscle myogenic amines. Here's where a lot of ours that we looked at: dopamine, norepinephrine, serotonin, histamine being one of those. Amino acids, GABA, glutamate, peptides, substance P, has to do with pain. Morphine-like substances, and enkephalin and endorphins. We'll get there. That's where we're heading. Here was Dale's law. A neuron makes the same neurotransmitter at all of its synapses. So one neurotransmitter for one neuron. Neurons are classified according to the neurotransmitter they release. For example, cholinergic, adrenergic, adrenaline, noradrenergic, dopaminergic, dopamine, serotonergic, serotonin. 2 important terms on this slide: agonism. So if you're taking a drug and it's an agonist, it's going to make that neurotransmitter work harder. It's going to make it more effective. It's going to promote it. If you're an agonist, you're promoting something. If you're an antagonist, obviously you're detracting from that. So some drugs that people take can make serotonin work less effective, or like we talked about with especially the opioid, the naltrexone. It's an opioid antagonist in that it blocks your cells from processing the opioid. It completely, and so somebody, like we said, pass out on a heroin overdose or an opiate overdose. If you spray that up their nose, it's gonna block all of that opioid action in the brain. Bless you, and it's gonna allow that person to immediately stop their overdose state. Drugs can do a lot of things. They can decrease the activity of enzymes that create or destroy a neurotransmitter. They can alter reuptake. We've seen that. Alter activity of a second messenger. Haven't seen that yet. Interfere with the operation of ion channels. We'll see that. Change the amount of neurotransmitter and neuron releases in response to an action potential. Cocaine, amphetamine. Drugs can block the receptors as well as the autoreceptors, so cells can't talk to each other. So the presynaptic cell may start making excess neurotransmitter. So there's a lot of ways that drugs can influence your natural processes. Acetylcholine, those are cholinergic synapses. Some drugs block acetylcholine, such as curare. Curare is found in the poison dart frog. So you think about the folks in the Amazon hunting the little monkeys up there with the blowgun, and they blow it, and it hits the monkey, and the monkey falls over very quickly. It's basically causing their heart and their lungs to shut down. It's counteracting acetylcholine. Myogenic amines. It includes norepinephrine, dopamine, epinephrine, serotonin. Get used to these. Get used to 5-HT-NE-EDA for dopamine. We'll use some of those abbreviations. Your text definitely uses those. So that's what those abbreviations mean. Tyrosine comes from food, used to make norepinephrine, dopamine, epinephrine. Talks about tryptophan. Anytime you see the word oxidase, we're talking about an enzyme. We're talking about an enzyme that's going in to eat things, to break things down. Neurotransmitters in this. So when we talk about a monoamine oxidase inhibitor, it's going to have multiple effects in the brain. So we'll get there with what that is. Sorry, we're going fast. I'm just trying to get through it. Biogenic amines continue. Amphetamines and other stimulants cause leakage of neurotransmitters to synaptic cleft, thus increasing the amounts of neurotransmitter in the gap when action potential occurs. GABA, involved in Xanax, Valium, is a universal inhibitory neurotransmitter for all parts of the brain. So it's going to make the brain work less. It's going to decrease the likelihood of action potentials. Does this by causing the leakage of chlorine ions into the neuron, thus making it hyperpolarized. Remember, it's taking it below the -70 resting potential. Anyway. Glutamate, the major excitatory neurotransmitter in the brain, works mainly by causing positive ions to leak into the neuron, hyperpolarizing it, making it easier to fire. Has a significant role in anxiety. Peptides, amino acids, protein molecules, opioid type peptides such as enkephalins or endorphins. Okay, we're coming in for a landing. We're on our last 2 slides. This is a oh gosh, why do I always have trouble remembering the terminology? Positron emission topography. Tomography, thank you. Now, this technology has been around a while. They're getting much more sophisticated. This was actually taken about 20 years ago. You can see the brain in its resting state, which areas are lit up. You can see when a person's listening to music, the occipital lobe, the frontal lobe, some of the parietal lobes, are firing. Somebody that's really cognitive, thinking a lot about possibly solving a trigonometry problem. A lot of the frontal lobe lighting up here. Visual, somebody that's really just focused on what's in their vision. Their occipital lobes lighting up. Language. Again, this is what we're bringing this back home because I want you to start thinking about geography, what we just watched, how everything functions, and what's going on in the brain. Language. Gosh, you see a lot lit up for language. It takes a lot of language is something very difficult. Using symbols, using our muscles of our voice, putting all these things together. Language is very complex. We're one of the very few species that has language. It takes a lot of our brain. It takes more than 10% of our brain to produce a language. Memory, a lot back here in the back regions. If you had your hippocampus, you could isolate that slice. You'd see that lit up. Auditory, kind of everywhere. Language and music together. And I can't see what that is. Okay, making sense of it all. Again, we're watching areas of the brain light up, thinking about all those action potentials happening, all that communication happening. Your acetylcholine system, this is in your text. This is on page 94. You'll notice that the acetylcholine system, it runs throughout the brain. There's a lot of the cortex, there's the thalamus, hippocampus involved. There's a lot going on in your acetylcholine system. If you look at the dopamine system, it's fairly complex. Remember we talked about what part of the brain is being activated when dopamine is firing? A lot of brainstem, a lot in here in the limbic system, in the midbrain, and some in the cortex, in the frontal lobe. Norepinephrine, again, you see a general, you see back here in the cerebellum, you see it having a general influence here in the cortex, hypothalamus, activating. Hypothalamus would be restoring. And then serotonin, again, the serotonin system, you see it, serotonin does a lot. Back here in the occipital lobe, all over the place. So when we're talking about each one of these specific chemicals, these are parts of the brain that are lighting up when that chemical's active. And that is it. This was our most difficult class. Okay? Don't be scared away by this class. Again, the testable stuff was highlighted. We will have a review. So that'll help people to isolate what to study. Because this was a semester's worth of material, because literally we just had to bust through so we could get to basically the pharmacology. And this will start landing more as you see how different drugs work. Okay. Ponca City, thank you for hanging in there. Alva, thank you for putting up with the technical difficulties. I appreciate you guys hanging in there. Jennifer, Woodward, thank you for having a late one with us. We'll see you next Monday. Don't forget your journals. See y'all. Bye. Whew, we go collapse now.
I know, I was like, maybe I'll need one. Nope. Not in here.
Hopefully that was interesting. That's sometimes tough to keep people that aren't interested in it. Did it make sense, at least? Okay, okay, good. See y'all. Have a good week.
See you later,
Thank you.
See you next week.
Okay.